The origin of animals. Endless forms most beautiful. The kingdom Animalia originated from the evolution of multicellular organisms from single-celled ancestors called protozoa. This occurred some 600 million years ago and is fairly poorly understood. What is an animal? Animals are eukaryotic, multicellular organisms that are motile, heterotrophic, and reproduce sexually. The wide range of animal species extant today all descended from a common ancestor. The last common ancestor of all animals is known as the Ermatozoan. To gain a deeper understanding of the origin and morphology of the Ermatozoan, it is necessary to look at the closest living relatives to animals, their sister group, the choanoflagellates, a group of single-celled microbial eukaryotes. Choanoflagellates. Choanoflagellates have flagellum that propel them through water and create a water column to collect bacterial prey on the surface of the microvilli collar. This collar complex features a flagellum surrounded by a ring of microvilli. William Saville Cant, 1888, was one of the first scientists to study choanoflagellates. He noticed the similar morphology between choanoflagellates and sponges that also have a microvilli collar they use for feeding. This microvilli collar is also found in most animal phyla, for example in sensory epidermal cells. The collar complex is also absent from other eukaryotic lineages, suggesting the low probability of it evolving easily through convergence. It can therefore be inferred that the last common ancestor of animals most likely had collar cells and used these to feed on bacteria. Genomic analysis of animals and choanoflagellates shows that they have other similarities. For example, in genes involved in adhesion, such as cadherins and C-type leptins. This means that genes required for multicellularity actually existed before multicellularity did. Genes that make animals different from their Ermatozoan ancestor and sister group choanoflagellates include genes involved in regulation, such as Hox genes, and cell signaling genes such as Wnt, Frizzled, and Hedgehog, and many more. The Ermatozoum is multicellular, but how did multicellularity actually develop? The evolution of multicellularity occurred once in animals. It most likely evolved through the mechanism of clonal development, where there is cell division without the separation of sister cells. Clonal multicellularity can evolve through loss-of-function mutations that would cause cells to fail in completing cell division. This is how the choanoflagellate S. rosetta forms rosette colonies. They are adhered through intercellular bridges and extracellular matrices. S. rosetta can fluctuate between single-celled and colonial forms. The possible selective advantages of clonal multicellularity could plausibly include cooperative feeding, division of labour, resistance to predators, and the formation of a stable internal environment. However, the evolution of multicellularity may well have been a neutral event with no fitness benefit and reached fixation by genetic drift. It is still largely unknown. So, the Ermatozoan most likely evolved from a colonial choanoflagellate. It is believed the Ermatozoan had a simple epithelium and used collar cells to feed on bacteria. It is thought it was capable of apoptosis and that its cells could differentiate. It's also believed that this animal underwent gametogenesis to produce sperm and eggs that would undergo cell differentiation and gastrulation to form offspring. While the exact origin of animals isn't completely clear, it is evident that choanoflagellates are key to understanding the morphology of this first animal and how it developed.